everyone. Welcome to the UIC Bioengineering Virtual Symposium. We're very glad that you can make it. I know in some of your countries it's 3:30 a.m. right now, so we really appreciate you doing this in the middle of the night. Just wanted to give you a little bit of overview. This symposium is really for you. It's really meant to have you get a sense of what UIC Bioengineering is about, what kind of programs are available, and also what kind of labs are looking for students. So please, get as much out of this as you can. We do have breakout sessions. So the way we're going to do this is from this point until 7:30 p.m., so about an hour and a half. We're going to have overview of the department, overview of programs. Labs will give 5-minute spiels about what they're doing. And then at 7:30 p.m. US Central Time, we're going to go to these breakout Zoom sessions that I sent you links for. So you can kind of go in and out of these Zoom sessions and ask questions from different labs. We're going to have faculty there. And there's also a Zoom session for with just the UIC bioengineering current students whom you can ask questions, that you wish with regards to how do they like the program, how did they adjust to Chicago, et cetera. And we have people who are international as well as domestic. So you'll be able to see both perspectives. So without further ado, let me hand it over to Professor Tom Royston, who's the department head of UIC Bioengineering.
Hi, thank you very much, Salman. Thanks for spearheading this and putting this together. I'm going to go ahead and share my screen. I've got okay, can you guys see my screen okay?
Yes, Tom, you're good.
Okay, great. Let me put this in presentation mode. I'm going to give just a very brief, broad overview of UIC and bioengineering, and then I'm going to hand it off to Dave Eddington, our Director of Graduate Studies, to give some more specifics about the graduate programs. But since some of you are from Chicago, but many of you are coming from other places all over the world, give you a little bit of an orientation. We are University of Illinois Chicago in the heart of Chicago. This is a photo taken from one of our newer dorms. This is the tall building. There's the Willis Tower, or otherwise known as the Sears Tower. So we are literally right in the heart of one of the major metropolitan cities of the world. And we try to take advantage of that. UIC itself, University of Illinois Chicago, is a major research university. Overall, right now, it's over 33,000 students. This is both undergrad and grad. I believe probably about 10,000 of those are grad students. It is a full-service university, 16 colleges covering pretty much every area. And we are a major research institution. We have a lot of sponsored research. We are very technical. We do have liberal arts and other areas. We cover everything, but we do have a significant technical effort here across many different colleges, a significant amount of development of patents, new tech, and this is a slide I pulled from the Dean of Engineering, highlighting some of the interesting things that UIC has been a part of, including some of the work on the original Star Wars back in 1977. Believe it or not, supposedly we're the home of the birthplace of virtual reality. We overall, as a university, we get high marks in terms of our general affordability and quality. This is primarily looking here at the undergrad. I just giving you a sense of what you would be coming to. We are one of the most diverse institutions in the United States, and in diversity in the undergrad program, we kind of look at these different categories of Hispanic, Asian, Latino, African American, etc. And this is basically just showing there's no ethnic or race majority here. This is a very integrated place. This is what we envision, perhaps a lot of the United States, hopefully looking like maybe 40, 50 years from now. At the graduate level, we are diverse in terms of having about, I think Dave will be able to give the exact numbers, about 50% domestic students, 50% foreign students coming from all over the world. As I mentioned, we have, it's a full service university. Our department, bioengineering, is both a part of the College of Engineering and the College of Medicine. The College of Medicine is also the home of the largest medical school in the country. More specifically about our department, our program, we are one of the oldest programs in the United States in the field of bioengineering, founded in 1965, one of the first to receive funding from the National Institutes of Health. Just giving a snapshot of where we are now in terms of the how many alumni we have with bachelor's, master's, and PhD degrees from our program. Specifically, most of these are from our bioengineering program, but we also do have a PhD and master's program in bioinformatics, which I'm sure Dave will mention as well, which account for some of those MS and PhD alums. Some stats in terms of the size of the student population presently, the number of faculty presently. We have both core faculty and adjunct faculty. So, and I'll get to a little bit more detail on that in a moment. But just to kind of, again, give you a bird's eye overview, we do offer bachelor's, master's, PhD degrees, the master's and PhD in both bioengineering and in bioinformatics. And this gives a snapshot of the degrees awarded in our PhD programs and in our master's programs over about the past 10 years or so. Also kind of highlighting that it's, it is, we'd like to see there be a larger percentage women in the program, but it is pretty good for an engineering program in terms of the number of the percentage of degrees at the PhD level, master's level awarded to women. We are a program that covers kind of all, I would say, pretty much all of the different niche or specialty areas that fall under the bioengineering or biomedical engineering area. And I should mention our department a year from now will be the Richard and Lawan Hill Department of Biomedical Engineering. So we are undergoing a slight adjustment in our name, which we believe is more reflective of the fact that pretty much everything we do is focused, has a biomedical engineering focus to it. This is a snapshot that's probably about a year old of our core faculty. The most up-to-date information can be found at our website. But basically, this gives a snapshot of what each faculty, their kind of some keywords in terms of their areas. Not all of them are available tonight, and not all of them at present may be seeking or looking to bring on new graduate students, but you will have the opportunity to meet a lot of them tonight. And so these are core faculty, and I did mention that we have what we call affiliate faculty. These are faculty who, typically faculty in other departments or programs at UIC. A lot of them are from other departments of the College of Medicine or the College of Engineering. The majority of our graduate students, their primary advisor is one of our core faculty, but not all. Dave would have the exact numbers. It might be around 20% will have a primary advisor being one of our affiliate faculty. Invariably, most of our dissertation committees or thesis committees will have a mix of faculty, both from the core faculty as well as our affiliate faculty, because so much of what we do and what you would be involved in terms of research is multidisciplinary and benefits from having a broad multidisciplinary perspective, expertise on your committee, and we can provide that. I should also mention that a lot of our committees also involve faculty, not just from UIC, but perhaps also from some of the close by universities where we have a lot of research collaborations. We're very close to the Northwestern Medical Campus, as well as Rush University Medical Center, and also there is the University of Chicago, and a number of our faculty have active collaborations with researchers at all of those institutions. that is all I have. I want to give plenty of time for Dave to drill in with more details on the graduate programs. And Salman, I can take questions now, or if we want to kind of stay on time, I can, however you want to do it. But it's you can also send me questions via chat.
Yeah, I
Thank
think
you.
Given that we have a pretty tight schedule, let's proceed to Dr. Eddington's presentation. And then if people have questions, you'll have opportunity in the breakout rooms, as well as you can put your questions in chat, and I'll be happy to relay them to the relevant parties.
Okay, sorry, I couldn't find where my mute was. Okay, so to answer the one question, Professor Royston had about 25% of our grad students, or PhD students anyway, are supervised by affiliate faculty, and then the other 55% are with the core faculty. So my name is David Eddington. I'm a professor and the director of graduate studies. My email are my initials, dte@uic.edu, so please don't hesitate to shoot me an email to ask any questions. But I'll generally be giving an overview of the requirements and coursework and kind of the things that you would do in addition to the research that you'll learn from the other faculty here. And is it presenting okay? Or are you seeing the notes? Okay. Okay, so first I'll talk about the master's degree in bioengineering. These are the degree requirements taken from the Graduate College website. Essentially, you need 36 course hours to graduate. Each course is on average 4 hours, and so that makes 9 courses. And then so we have a coursework only and a thesis option. And so if you do a thesis, then you need 12 hours at the 500 level. Those are typically graduate college classes, but you can get credit for any course 400 or 500 level. And then if you're doing the non-thesis, then you need 16 hours of, let me get a pointer out here. There we go. Okay, so if you're doing the thesis, you need 12 hours of 500 level, non-thesis, you need 16, and otherwise they're the same. The only required course is that you take Bioengineering 595, which is a seminar course, and then we have some other requirements. A certain number of these hours must be in bioengineering department, but other than that, the rest can be in any other STEM department, and so you can really tailor fit your master's coursework based on your research interests, because bioengineering or biomedical engineering is a large and broad field. Okay, oh, and if you're doing a thesis, you need a certain number of thesis hours. For the PhD, you need 108 total hours. Of those, 48 need to be coursework if you're coming in without a master's degree. If you're coming in with a master's degree, then only 24 hours of coursework. And then a similar number of requirements. A certain number must be bioengineering courses. A certain number must be 500 level. And then you also need to take seminar 595. And there's a few other courses that we strongly encourage our students to take that are statistical basis and quantitative based courses that I won't go into here. And then the other 60 hours are your thesis research. And then for the master's in bioinformatics, there's a, oh, I didn't share. Okay, I'll just stay on the presentation. There's a link here, but I want to keep everybody on time. I don't want to be going on. So there's a few, so for the master's degree in bioinformatics, the coursework is a little more restrictive, and you have to pick a certain course among others. And this is a really long web link, and I apologize for that, but it's navigatable through our department website in the graduate and the master's in bioinformatics. You should be able to find it, and if you can't find it, just shoot me an email and I'll send it along. But essentially it's the same number of hours and everything. There's just a few more required courses. And then for the PhD in bioinformatics, the only one of the differences, so they have a little more 500 level requirements, and they do also require a qualifying exam for bioinformatics. So you take your coursework, you do a qualifying exam, and then you do your prelim exam, and then you do your thesis. For the PhD in bioengineering, we do not have a qualifying exam, so you do your coursework, you do your prelim, you do your thesis. Okay. So there's a lot of courses available. I just listed some here, such as human factors, engineering for medical devices, MRI in practice, biomedical computer vision, nanobiotechnology, biotransport, engineering, numerical analysis, and multi-scale modeling. And these are all bioengineering courses, and that's not to mention all the other courses. And there's a master list shown here. I'm just gonna click on this. There we go. Okay, can you see my web browser now? Yeah, okay. So on the departmental website in Graduate, if you click on courses, this is where I am. And then there's this link here, Bioengineering Graduate Course Electives, is a link that of every single course bioengineering graduate students have taken. And so this is just a really exhaustive list of all these different courses. So you can specialize in computer science, mechanical engineering, entrepreneurship, other biomedical science courses, and all across the board. And so the coursework is very open-ended, as long as your thesis committee, if you're doing a thesis, agrees with it, or you're taking STEM courses. Okay, so let me get back to my main presentation. Okay. And then in addition, the PhD has an annual evaluation, and I put a link here as well, but I won't go to this link. But essentially each year you meet with your committee and you present your progress and plans to your PhD committee, and this ensures that you're making slow and steady progress and everybody's on board, so there's no surprises at the end of your studies, and that you know what you're getting into and what is expected of you to clarify these things. And lately, usually these are done in person, but lately we've been doing them on Zoom, and it's not too bad. So we have that as well for all PhD students. The preliminary exam, that's for both bioengineering and bioinformatics. Again, here's a link to more details, but it's ideally done in year 2 or 3 of the PhD, and it includes both a written and an oral component. And the written component is essentially a 7-page grant proposal on your thesis topic that you then present in a 45-minute-ish presentation to your thesis committee. And then the qualifying exam is a written qual for bioinformatics, but not for bioengineering. And that is all I had. Again, please email me. My email is dte@uic.edu, and I can take a few questions, or I got us back on time as well.
So thank you, Dr. Eddington. Dr. Eddington's email address, as he said, is very simple, dte@uic.edu, and he's very helpful with guiding you through this process. So please do email him with these specific questions. So, for over the next 30 minutes or so, what we'll do is we'll have different labs give 5-minute overviews of what they're doing, what kind of research that they're engaged in. Then we'll have a 10-minute break, and we'll do it again in part 2. So without further ado, the first lab that'll be presenting is Dr. Karman's lab. So Dr. Karman, if you can please share your slides,
Sure.
much appreciated. Thank you.
Of course. Do you see them all right?
Yes.
Okay. Thank you. Hello, everyone. My name is Müge Karaman. I'm a research assistant professor at the Department of Bioengineering, and I've been conducting my research at the Center for Magnetic Resonance Research of UIC. So CMRI, in short, has 2 locations at the West Campus, one at the Advanced Imaging Center, which houses a 3T fully research dedicated MRI scanner. The other one is at the hospital outpatient care center, which has a 9.4 human research scanner. My research is primarily at the 3T scanner site. Also, UIC has a rich environment for MR research with a total of around 31 full-time faculty members, such as Dr. Tom Bryston and Dieter Klatt from our own department. So MRI is a technique that uses a powerful magnet and radio waves to create detailed images of the internal body structures. The first step in this pipeline is MRI data acquisition, which produces a set of MRI images, either anatomical images that the radiologist can interpret directly, or other types of images based on more advanced MRI techniques that have functional information, such as diffusion MRI. Unlike the anatomical ones, these types of images need to go through an additional step for MRI data analysis to produce MR-based imaging parameters. These images are then evaluated by a team of clinicians who then make important decisions such as diagnosis, treatment, or prognosis evaluation. At the CMRR, research for MRI data acquisition is primarily led by Dr. Zhou Zhao, who is also affiliated with the Department of BIOE. My contributions are mostly on the MRI data analysis part. These two research areas, however, are intertwined, and all of the center members are substantially contributing to both areas. Besides our current and past members, we are collaborating with many scientists and clinicians at UIC and other institutions from various departments ranging from radiology, neurosurgery, oncology, pathology, to computer science. The main goal of developing advanced techniques for MRI data acquisition is to improve the image quality in MRI. Today, I'm going to show a few examples of our studies in this area. One of the aspects of improved image quality is to reduce geometric distortion, which is inherent in many conventional MR data acquisition techniques. Our laboratory is active in developing advanced techniques to reduce such distortion, as the one that you see here, where we were able to produce substantially improved MR images in these distortion-prone regions. Another area that the current MRI techniques require improvement is to improve spatial resolution. In one of our recent studies in which we applied a novel 2D-RF pulse design, we were able to improve the spatial resolution up to 0.6 millimeters square and demonstrated this technique to resolve the structures and substantia nigra of the Parkinson's disease patients. Another very important challenge in MRI is low temporal resolution, which prevents MRI to capture rapid dynamic events. In our most recent work, we were able to produce MR images with sub-millisecond temporal resolution, as demonstrated here on cardiac MRI. Another area that the center specializes is in MRI data analysis, primarily for diffusion-weighted MRI data. Diffusion-weighted imaging is mainly used by the clinicians to non-invasively characterize the complex biological tissue, such as the cancer tissue, as it provides an opportunity to reveal the underlying microstructural information by measuring the random motion of water molecules within the tissue. In a typical MRI experiment, the collected MRI data for each voxel in the image is a signal intensity curve that decays with increasing p-value. What is challenging here is to model this signal decay curve to produce interpretable parameter maps. One area that our research is to develop mathematical techniques to analyze the diffusion MRI data to produce maps that reveal additional tissue property-related information, such as tissue heterogeneity, compared to the conventional techniques. So we investigated the feasibility of these advanced techniques in a variety of clinical problems, such as pediatric brain tumor grading, where we see significant differences in model parameters between the low and high-grade tumors. And also in grading adult gliomas, I want to mention here is that non-invasive grading of brain tumors is of particular importance, since biopsy can be prohibitive because of the significant risks associated with the invasive procedure in the brain. We also investigated these techniques in classification of different types of gastric cancers, which can be helpful for the clinicians to determine the most favorable treatment strategy for gastric cancer patients. With this technique, we also characterized breast cancer, which is the most common cancer diagnosed in women in the US, and one of the cancer types which is curable given the accurate and timely diagnosis. Besides these classical techniques, we are working on machine learning-based analysis of diffusion MRI data. In this technique, for example, we developed a multitask deep residual neural network for prostate cancer detection. This technique enables the analysis of the MRI data without relying on mathematical models and its assumptions, but instead learning from the decay pattern of the diffusion signal. We have also recently developed a machine learning-based technique to analyze the model-based diffusion parameters for breast cancer characterization and tumor response prediction. We also have some earlier studies based on these techniques in cancer and other types of diseases. That's all what I have today. Thank you for listening. I also would like to thank the many people who made this work possible, our collaborators, center members, and also the funding sources.
Thank you, Dr. Karaman. And again, Dr. Karaman's information is on our website, bioe.uic.edu. So please do contact her if you're interested in MRI research. So our next speaker is Dr. Linger.
Okay. And can you hear me well?
Yes, very well.
Okay. So my name is Andreas Linninger. I would like to introduce current activities and point out mainly the research that we do in the physical brain and also in drug administration to the central nervous system. So our focus is exploring diseases of the brain and the central nervous system, which is motivated by the large number of people that are affected by some imbalance in metabolic functions or blood supply, but where, despite the significance of the diseases, we have not really obtained a very good understanding about the reason why the brain stops functioning. So Parkinson's disease is one example that affects at least 500,000 people in the United States, which is related to an upset in the dopamine metabolism. Alzheimer's disease is associated with nerve cells breakdown, and it affects millions of people in the United States and around the world. So our goal is to help understand the causes of the disease that affect cerebral metabolism and blood flow. One of the projects that we currently pursue is to dive in the microvasculature of animals and of human brains. Here you see a picture of 2-photon image acquired microcirculatory networks for the mouse. And we like to look at the blood flow and the substrate exchange across the blood-brain barrier using these 2-photon imaging data. But in addition to observing quantities in small capillaries and arterioles and blood vessels, we are also looking into analyzing the relationships between flow, pressure, and solid transport by using mathematical modeling techniques. So on the right here, for instance, I show a mouse brain, but this mouse brain has been generated on a computer using morphometric parameters that we acquired from imaging. Here it gives you a little bit of an overview of the terrain that we are working in. We use at the small scale multi-photon imaging data sets that basically create an inventory of all the blood vessels, including capillaries at the smallest length scale. But the problem with these small samples is that if we want to perform blood flow computations, the boundaries are cut off and the blood vessels are cut off where the image view ends. And that affects our ability to simulate blood flow in these small samples. So what our lab has been doing is we have created network structures that emulate the morphological parameters of our image sets, but extend them to larger sets. You can see here one example that is more than 3 millimeter. And we're able to create on the computer an entire mouse brain with the connectivity down to individual capillaries. So we name that the complete cerebral circulation in mouse, where we can then study the effects of, for instance, blockage. Here's an example which shows different images for increasing amount of blockage that is caused by lymphocytes. And we can see here very conceptually at least how the flow goes down from initially red, which means good perfusion. And as you get more and more blockage of the capillaries, you have more reduction in blood flow. We also look into the control functions of the brain and pathologies such like microinfarcts. We would like to know not only blood flow changes that occur at single vessels, but at the whole network. The second area that is also related to blood flow is at a different length scale. This is blood flow in larger vessels. Here we see an arterial tree that's acquired with MRI imaging and with CNA phase contrast MRI, which we use to obtain blood flow measurements. And our goal is here to analyze blood flow and hemodynamics across all length scales. Here, for instance, I see an example of a video of a colleague, David Barlow, who shows that the blood flow at the micro level is not actually a single fluid, but it's a suspension of red blood cells and plasma. And that gives rise to complex flow phenomena, which we study both with imaging techniques, but also with simulations. Applications include, for instance, the risk analysis of cerebral aneurysms. Here you can see a network that has been reconstructed from a subject that we have imaged, and we have created a computer model of the blood flow through the arterial network here and the aneurysm. Here we want to explore how the blood flow condition changes that occur in these pathologies affect the distribution of blood flow and also the risk of rupture in such aneurysms. Here, for instance, is an example of such a virtual endovascular intervention. So there's a stenosed vessel, which means that the blood flow is reduced due to the reduction of the diameter. The common treatment includes putting a stent that widens that vessel and then restores blood flow and also conditions on the arterial walls that may lead to problems. Here we have, before the intervention occurs, conducted a fluid mechanical studies, a hemodynamical study as to what would the expected post-treatment flow rate look like. So here we have the stenosis and the velocity profiles through computations. And here we have the expected results that occur when you widen this stenosed vessel using three-dimensional CFD simulations. Finally, we are also interested in the administration of drugs to the central nervous system, because the blood-brain barrier prevents drugs from reaching the brain tissue, and therefore many therapies are ineffective because they don't really reach the brain. So we want to overcome the barrier that is posed by the blood-brain barrier using invasive techniques. These invasive techniques include intrathecal injection. Here it's not a real patient, but a three-dimensional functional model of the central nervous system, which has the spinal cord that is filled with CSF and the brain. And in this fluid, this is called the cerebrospinal fluid, we inject catheters and watch the dispersion under the pulsating flow of cerebrospinal fluid. So in this work, we perform imaging, but as you can see, we also perform 3D printing so as to being able to study the fluid mechanics in models that focus on the flow and are easier to obtain data. One application here is the so-called magnetic drug targeting. For instance, if you administer a drug in a body, you have the problem that it goes everywhere, but also may not really reach the brain. If you want to treat a lesion in the spine, we propose to use a magnet, and the magnet localizes the drug in a small region where it is desired to operate. We do that with supraparamagnetic nanoparticles. You can see an image of a nanoparticle solution. When you place the magnet, you can see how these magnets attract the nanoparticles. And by virtue of the magnet, we are able to control where the nanoparticles are active. And especially for chemotherapies, we may be able to localize the effect just in the tumor site, but not disperse it through the entire central nervous system, where the chemotherapy can cause severe damage. We also have studies going on in which we do in vivo experiments in animals. This was a collaboration with people at Biogen. Here we did a study where we infused drugs into a cyno monkey, and we then compared our predictions with the actual drug dispersion using PET imaging combined with PC-MRI. So in this project, we are basically trying to bring the experimental work in the lab, the simulation work in the lab, into the industrial practice for intrathecal drug delivery, where drugs are injected in the spinal cord and then dispersed in the central nervous system. The skill sets that our students in the laboratory develop include biomechanics, mathematical modeling, and interpretation and analysis of MRI flow measurements that either we perform in collaboration with the imaging center, or in collaboration with groups that we work together with. Some of the highlights that have recently been selected for journal covers are shown here. We are a group that works in different areas. We call that interdisciplinary work. So we have publications in neurosurgery, oncology, cerebral blood flow metabolism, and we try to bring a lot of our work into clinical practice or into neuroscience. Finally, I would like to show the host of people which we are privileged to work with. We try to work with people who are specialized in imaging or who have capabilities that the lab doesn't have. But the students in the lab have the opportunity and the challenge to collaborate with people around the world. I thank you for your attention. I've put my email here in the top of the slide in case you want to see me in the breakout session. And I hope you enjoy the session. And I thank Salman for organizing this workshop.
Thank you, Dr. Langer. That was a very, very nice talk. Our next speaker is Dr. James Lee. Dr. Lee, the floor is yours.
Thank you. You see my screen right now?
Yes.
Hi, everyone. I'm James Lee, the Director of Cell Mechanics and Signaling Laboratory. The mission of our research group is to apply biophysical engineering principles and techniques to investigate the mechanical and chemical processes in neurodegenerative diseases, especially Alzheimer's disease, AD. Ultimately, the information derived from our research will provide insights into the mechanism underlying the pathology of the disease, early diagnosis, and therapeutic strategy. By focusing on Alzheimer's disease, the increasing number of AD victims puts a heavy economic and emotional burden on society, and AD has become an urgent national health and research priority. Our work focuses on investigating the mechanism and the role of mechanical alterations in AD brains. Our paper do get some attentions as reflected by the number of citations. Here are a few examples. We focus on microglia, astrocytes, neurons, and the blood-brain barrier. Specifically, we study how oxidative stress, amyloid beta peptide, and tau affect cell function. So let me give you a lab tour. Our lab is located in the College of Medicine research building. So going through the front door of our lab, you will see a microscope room on the left, a cell culture room on the right. Going further, you will see a wet lab. In the microscope room, we have a Nikon inverted fluorescent microscope. This setup looks very simple, but it allows us to do some interesting quantitative observation. For example, we can remove cholesterol. And directly observe membrane domain to become more molecularly disordered. We can also increase oxidative stress in the cell, and and membrane domain becomes more molecularly ordered. Also, we can use molecular rotor to probe membrane fluidity and to observe the membrane fluidity. At the subcellular level. We also have an atomic force microscope. This heavy metal instrument allows us to perform mechanical and structural
Looks like we lost Dr. Lee. Dr. Lee, can you hear us?
Are the AFM images of an amyloid beta oligomer and fibrils. Are you guys hearing me?
Yeah, you went away for a little bit, for like a few seconds.
Yeah, it was not stable, the internet. Okay. Here is our cell culture room equipped with biosafety hood, CO2 incubators, and liquid nitrogen tanks for cell cryopreservation. Our wet lab is equipped to perform animal dissection, cell isolation, purifications, and purification, quantitative measurements for proteins and gene expressions, immunohistology, and productions of elastic substrate. Thank you, and we are now recruiting new PhD students, and you're welcome to join us. That's it.
Thank you, Dr. Lee. What we'll do, we're a little bit behind schedule, but it's around 7 p.m. Chicago time, and we'll take a 5-minute break until 7:05, and then we'll begin part 2, which will be another 6 professors providing their lab's research. If you do have any questions for the professors who have already presented, please feel free to put that in the chat, and they're monitoring the chat as well. They can answer the questions, but otherwise we will have a breakout session after this main event as well. So 5-minute break, and we'll be right back. Hello, welcome everyone. Welcome back, I should say. So let's start our part 2 of the overview of bioengineering labs. And our next speaker is Dr. Strocho.
Okay, thank you. So I want to tell you a little bit about the Nanoengineering Research Laboratory. We have a history of integrating man-made nanostructures with biostructures and using Raman spectroscopy to identify biomolecules, many associated with diseases. These man-made nanostructures include semiconductor quantum dots, nanowires, including graphene structures and carbon nanotubes. We look at both optical and electrical operational modes. The optical is well suited if we want to place nanosensors inside of cells and monitor concentrations of analytes remotely. Recent placements, some recent placements are listed here. Both industry and universities, and our funding has been from a variety of sources. At the current time, we are seeking students who are interested in master's research, but that could change in a couple of months. So this is my contact information, and I'll repeat that. So illustrated here are several different nanosensors. The upper right is a molecular beacon where we have a quantum dot that's linked to a nano quencher by a DNA molecule that binds selectively. This DNA molecule is an aptamer. It binds selectively to an analyte the same way that an antibody does. The lower right is a transistor structure where there's an aptamer functionalized to the surface, and on the end of the aptamer there's a molecule methylene blue, MB, which is electron rich, and when it binds to the analyte, that there's a conformational change in the DNA aptamer, and one can get a current between the source and the drain. So they're both electrical and optical modes. This shows an example that we have extended. It was originally Alan Heeger and Kevin Plaxco. You may know Heeger. He's a Nobel laureate for conductive polymers. This shows an aptamer near a gold surface with a methylene blue on the end. When you introduce a molecule, here platelet-derived growth factor, that binds to this aptamer, you can induce a conformational change that causes an electron to be done to the substrate. So it's an electrical sensing mode. We've also used quantum dots to stimulate ion channels. This shows horizontal cell from catfish and retinal horizontal cell, and quantum dots are placed in suspension near the bilipid layer. There are various ways to gate these ion channels, mechanically, chemically, electrically. Here we're considering electrical gating by a quantum dot with a dipole moment in close proximity to the ion channel. This gives an example of using a quantum dot-based sensor to detect calcium in the cytoplasm. This is one of the first cases where such molecular beacons have been introduced into cells. This is another example of detecting TNF alpha in the cytoplasm. We also do Raman scattering using SIRS, surface-enhanced Raman scattering. As you know, the SIRS works on the principle that if you have a nanostructure, a metallic nanostructure, the incident electric field produces a plasmon, which has nearly the same frequency as the incident laser radiation, but you can get fields that are 10 to the 7 times stronger than the laser. So effectively, the plasmon amplifies the incoming laser signal. In the laboratory, we have Raman capability, photoluminescence, a scanning tunneling microscope, atomic force microscope, an inverted Nikon, and a metallurgical Nikon, and other items such as gel electrophoresis. This shows more on the quantum dots near ion channels. If you're interested, please contact me. Thank you very much.
Thank you, Dr. Storcho. So our next speaker is Dr. Yao. Dr. Yao, can you hear us?
Yes.
Okay, perfect.
Let's see. Can you see my screen?
Yes.
Okay, great. Yeah, I will do a brief introduction for our lab. Our lab is the biomedical optic and functional imaging laboratory lab. Our lab is working on the custom design optical instrumentation and also using clinical imaging instrumentation. But for all of this study are focused on the retinal part. That means the back of the eye. We are working on confocal imaging system, OCT angiography, and also funded camera system, for the eye research, including both animal model and the human subject. The retina is the special biological, tissue at the back of the eye. From viewpoint of neuroscience, the retina as a cell belong to, to our brain. That means it's a central nervous system. So the retina is a very complicated neural network. So as can affect not only the eye disease detection or management, and also some people believe it can also work as a window for the study of the brain, like dementia. So currently, there are some, technical limitation, like the image field of view, resolution, contrast, and also, the functional information correlate with the morphological, structure of the retina, because again, this is a very complicated neural network. So our lab is working on a few projects, try to, do some contribution, to overcome some of the technical limitation for better, eye disease detection and treatment assessment. One challenge, for the retinal image is the limited field of view, because in traditional funded camera, both the illumination light and image light are delivered from the pupil. That's a limited field of view for each image, just to a small portion of the retina. But the retina cover, from here to here, that's more than half of the eyeball. In principle, it's also possible, to deliver the light outside of the pupil. That's from the scleral area. People call it the pars plana. This area, can provide relatively better optical transparency compared to other, scleral region. Using the pars plana illumination, we were able to demonstrate, ultra-wide field founted image. This is our first demonstration using a baby camera, for newborn baby image. Newborn baby image is very important for, for our P screening. That's in prematurity disease. So from the picture, we can see we already be able to reach the far end. That's the oral serrator of the retina. So this is, comparative study using a clinical device, RedCam, compared to our prototype. So this is demonstrated feasibility. Recently, we also are pushing this, for adult version image. That's contact-free image based on standalone system, and also, portable device, for DR screening. This technical demonstration had been, validated, but to push the technology, for useful clinical device, it's still, a long way to go to optimize the system. That's why we need, more lab members to join us to push the technical development further. We are also working on the resolution improvement for the retinal image. So this is our recent demonstration by using a virtual structure detection system to enhance the resolution, to achieve single cellular resolution to separate rod and, cone photoreceptor from each other. This is from in vivo, from awake human subject. We are also using animal model, try to understand the retinal neurovascular complement defect, correlated with the eye disease. RD10, that's, photoreceptor degeneration mouse model, we can see the vascular defect will occur correlated with photoreceptor degeneration over time. Also, we can use the OCT, OCTA, to study the eye development. So that's the haloid vascular structure only occur during the retinal development. That's after the birth, for example, for matured baby, we cannot see the haloid vascular structure. But if it's the baby delivered premature, some of them we may observe the haloid vascular structure. Another project is the clinical data processing in collaboration with the eye doctor. We are also working on the clinical data processing. That's using machine learning, deep machine learning, for digital data processing of the retinal image, and also for the disease staging classification. Okay, that's basically our lab activity and project, and I will be happy to answer any questions later.
Thank you, Dr. Yao. And again, Dr. Yao, as well as other faculty, are going to have breakout sessions in the next 10-15 minutes or so. But let's go on to the next presentation, which is happens to be me. Okay, I'm going to keep it fairly short and sweet, because I want to make sure we have enough time for the remaining presentations. But my lab is called the Microfabricated Tissue Models Lab, and I have been at UIC for 5 years, and I'm an associate professor. And so what we do in my lab is we engineer 3 kinds of tissues currently. We engineer liver tissues, cardiac tissues, and intestinal tissues. And when I say we engineer tissues, what I mean is that we take cells from people who have passed away, or stem cell derived cells or cell lines, and we put them in various biomaterials, and then we try to figure out how do we maintain their functions outside of the body. This is what I mean by engineering tissues. So that's one major engineering part of my lab. The other part of my lab is to then utilize these tissues for various applications. So the applications are cell-based therapies, where a patient is suffering from organ failure and they need a engineered tissue to keep them alive. The second application is to model various human diseases for the development of new therapeutics. So think about like COVID-19, for instance. You know, we don't work on COVID-19, but it's a good example where in order to develop new vaccines and new therapies, that disease has to be modeled in vitro outside of the body so that therapies can be screened on that disease. And then finally, we utilize these tissues to do a drug metabolism and toxicity screening so that these drugs don't have adverse effects. This is kind of the overall viewpoint of my lab. Let me give you a couple examples. So one example is we utilize microfluidics on a small chip to basically generate very reproducibly sized three-dimensional microtissues, as we call them. So we have a device where we have oil coming in from one direction, and then cells and, let's say, protein coming in from the other direction. And because oil and water don't mix, we get these droplets with cells in them. And then when we heat these droplets, they polymerize into these miniature microtissues. And then we can place these microtissues in various microwells. We can put other cell types on, and we can generate these very precisely sized tissues for various applications. Here's sort of a video just to kind of give you a sense of how it works. If I can play the video, there you go. So here you see, we're looking at the microfluidic device from the top, and you can see these very reproducibly sized microtissues that are shooting out of this device at a rate of 45,000 microtissues per hour. So the other thing we do in my lab, just to give you an example, is we mimic human diseases. So as you may know, fatty liver disease is becoming a huge epidemic globally. So people are getting obese, they're drinking too much alcohol, and these things end up causing the liver to become fatty. And when the liver becomes fatty, when it starts accumulating fats, it can lead to diabetes, fibrosis, and liver cancer, for which there is no current disease. So here's an example where we take our liver cells. These are liver cells that are surrounded by fibroblasts. And when we put low glucose, these liver cells don't become fatty. But when we put high glucose, you can see that this green is basically the liver is becoming fatty. And when we look at this fatty liver, it has many features that are representative of the clinical situation. And one can then use this for drug screening or to develop new therapies. Another example is we utilize biomaterials, various biomaterials, to construct a three-dimensional organ for therapeutic purposes, so that if somebody is suffering from organ failure, the therapeutic tissue could be implanted in that patient. Here's an example of us utilizing silk. So the ancient material silk, which some of you may have sported on as clothing, but it turns out that silk also is a very biocompatible material, and we can create these silk scaffolds. We can put liver cells or cardiac cells or whatever you name it, and we can create these three-dimensional tissues which can then be used for implantation and to provide therapy. Now, we are not quite at the clinical stage yet, but we're definitely getting there very quickly. So my lab is as I mentioned, it's called the Microfabricated Tissue Models Lab. This is the website of the lab, and it's a very inclusive lab. As you can see, we have people from all over the world. I think last time I counted, there's 9 or 10 languages that are spoken or can be spoken in the lab. Obviously, we all speak English, so we can understand each other. But this is something we greatly pride ourselves on, and we greatly pride being at UIC. So if you are, no matter where you are in the world, I believe UIC can be quite a nice fit for you. And then where do our alumni end up? As you can see, that people end up for a postdoc at University of Pennsylvania or in European countries. And we have placed several students in industrial positions at biopharma, medical device industry, and also startups. And then we're very lucky to be at UIC with wonderful scientists and wonderful collaborators. We collaborate with people at UIC as well as across the nation, as well as across the globe. and we're funded by the National Institutes of Health and the National Science Foundation. So I'm going to end here, and what I'm going to do is that in the graduate students breakout room, 2 of my graduate students are in that room. If you have any very specific questions about my lab, they can address that, and we have other students in that breakout room as well. So thank you for your attention, and I will go ahead and move to the next speaker, who is going to be, as soon as I stop sharing, I'll be able to find that out. So the next speaker is Dr. Ma. Dr. Ma, the floor is all yours.
Thank you. So I'll give a brief introduction for the research in my lab, which is a computational lab, basically. So if you have questions, please drop me an email. So the overarching theme of my research is to uncover the physical principles that govern the behaviors of complex biological systems. Such systems like, for example, a cell, they typically has multiple functional layers, right? Like proteins or cell modules. The behaviors of a functional layer is fundamentally different from those of its building blocks. That's because of the complex interactions between the building blocks, and these are governed by the physical laws. So if you want to understand how a biological system works, you have to have a physically rigorous mechanistic model that can reconstruct the behavior of a functional layer ground up using the behaviors of their building blocks and physical laws alone, without applying any kind of biological assumption that cannot be explained by physical law or actually violates physical law. So there are 2 major directions. One is we want to understand the molecular mechanism of activity dynamics in proteins. Another is to understand the mechanism of the cytoskeletal microtubule network. So first, active dynamics of proteins, right? Proteins are the basic functional building block of a cell. And they pose many interesting questions like, how does enzymes work? How can allostery happen? And why some mutations can alter protein functions whereas others don't? And how to design artificial enzymes? And why proteins need to have a specific structure, right? And of course, malfunctions of proteins leads to so-called molecular diseases. So the basic thing is that all the functionally important processes in proteins are activated process. For example, this opening of the HIV protease, basically this kind of process, it needs to cross a energy barrier that is much higher than thermal energy. So how we want to understand how do proteins do this? And our basic theoretical tool is the energy flow theory developed in our group, in which we basically can study the potential energy flows through individual coordinates and also between different coordinates, and also the kinetic energy flows through different coordinates. So here is an example for the so-called flap opening process of the HIV protease, right? You can see here are the potential energy flows. So basically what we can see is that almost all the backbone dihedrals have very high potential energy flows. That means the dynamics is very global and collective. And also many dihedrals of the non-active site residues with drug-resistant mutations, they have high energy flows. That means they are essential for the flap opening process, which is critical for the functioning of HIV protease. So basically mutations in them will disrupt this energy flow and consequently inhibit this flap opening process. And that's why mutations in them produce drug-resistant effects. So the basic insights we gain from this type of study is that basically you can see potential energy flows will flow from fast, slow coordinates in activated processes. And kinetic energy is actually the dominant mechanism for the energy activation. And also the energy flows within the protein, they follow a hierarchical structures. So then why does the protein want a structure, right? In our opinion, the protein has a structure in order to build the proper energy flow channels to deliver energies to the reaction center or the active site so that they can accelerate chemical reactions efficiently. Next thing is the microtubule network. So microtubules are the backbone of the cytoskeleton, and they are essential for many cellular processes like mitosis, morphogenesis, polarization, differentiation, transportation, etc. And you can see the microtubule network in different cellular processes or different cell types takes very different shape. And because microtubules are so important for so many processes, right? Malfunctions of microtubules will basically lead to malfunctions in cellular processes, which naturally leads to disease. For example, if you have microtubule malfunction in mitosis, then you lead to aneuploidy. Basically, the genetic information of the new cells are all messed up, which either leads to cell death or cancer cells. And also malfunction of microtubules in neurons will lead to neurodevelopmental disorder or neurodegenerative diseases. So it's very important to understand how microtubules function. So all the functions of microtubules are driven by its assembly dynamics, which is called dynamic stability, which is the spontaneous and stochastic switching between periods of continuous growth and shortening. So here is a short movie of microtubules in a cell, and you can see basically microtubules keeps going growth and shortening, growth and shortening. So in order to understand how microtubules function, you have to understand the physical mechanism of this dynamic instability. And what we found is that dynamic stability is driven by the conformational change of the building block of microtubule, right? Microtubule is really a tube of tubulin dimers. So here is the atomic structure of a tubulin dimer, and it undergoes conformational changes, which is coupled with GTP hydrolysis. But tubulin dimers, they need to do these conformational changes within the two-dimensional lattice of microtubule. So they are subject to different constraints due to the different lattice environment. Therefore, they need to follow different kinetic pathways, which leads to different phenomena of dynamic stability. So basically, we consider these tubulin conformational change and the lattice geometry, and also based on the laws of chemical kinetics, we can construct a rigorous structure-based mechanical model for dynamic stability that can explain all the phenomena of dynamic stability. Based on that, then the next questions we are going to ask are basically what are the molecular mechanisms that individual microtubule-associated proteins can modify the behaviors of dynamic stability via protein-protein interactions with tubulins. And also, how do different microtubule-associated proteins, they cooperate with each other in cells to achieve the desired behaviors of microtubule dynamics for different cellular processes. So this is the kind of work that we do in our lab. With that, I want to thank you and give it back to Professor Katani.
Thank you, Dr. Ma. Our next speaker is Dr. Patton. Dr. Patton, the floor is all yours.
Okay, thank you. Let's see, share. Hi, everybody. I'm going to zoom out, as we might say. I'm very interested in much larger scale things. I'm interested in biomechanics, but only because it's fascinating for the challenge that it is to the brain and what it takes to move. And I would say to those of us that are out there that don't believe that the nervous system is important to movement, I would say that the nervous system is actually probably evolved and actually created by the need for organisms to develop and move. And that's really why nervous systems have evolved. So you can tell I'm a motor chauvinist in my thinking. But so I'm kind of on the fringe between, maybe the way to say it is the neuroscience or neural engineering and mechanical engineering that's necessary in order to get things done. I'm also fascinated by this idea that humans interact with each other and with machines and through machines with each other all the time. So this is an interesting frontier that's becoming a bigger and bigger deal every day. And I don't even need to say that to most of you. You realize that as soon as you pull out your phone and you realize that you're using machines more and more every day. So studying how we interact with machines is great. I'm very interested in also the idea of rehabilitation and learning in particular. The idea that when we've had an injury, very often we have to learn how to move again or learn how to deal with things again. So this is how I work. I like to use robots, and I'm at the best place, I think, for the research on such a topic. This is at the Shirley Ryan Ability Lab. The Shirley Ryan Ability Lab used to be called the Rehabilitation Institute of Chicago, and it's world famous for its research on the science of rehabilitation. So, and I should say you can reach me at these addresses up at the top on this slide. So I have a lot of interaction at the Ability Lab, the Shirley Ryan Ability Lab across town, and that's where I do most of my work. So what do I do? Well, it's a lot of work that has to do with training, but I'll say also that my laboratory environment is integrated into the clinic so that I run into patients, therapists, and other types of clinicians, physicians, nurses every day, and I know what the real life challenges are. Okay, so this maybe a simple example of what we can do, but the real question is what and how we accomplish things. A lot of times what it is it's very open-ended. We have a robotic device that this person is holding on to, and this is a virtual reality device called Paris that was developed here at UIC in the virtual reality group. But this person's interacting with fake stuff in first person using stereo glasses so that they see it in first person and actually feel it as well. The robot renders the sense of touch. We call this haptics, and you can create any kind of environment and have all kinds of fun with these types of things, but most importantly, you can do good science. You can ask questions like, how does the brain react to disturbances when we're moving, and how do we keep stable? And most importantly, how do we recover from neural injury after we've lost our ability to move from, say, a stroke or a spinal cord injury or multiple sclerosis, traumatic brain injury, a lot of other diseases. So there's a lot of different ways to do it. And we draw a lot on neural computational approaches, neural network ideas, and also we're heavy on predictive modeling simulations of how you execute movements. And I think, there's a simple example I can show you. We have one of our applications is the idea of error augmentation. This is the idea that if you make mistakes, and you learn from your mistakes, then why not artificially make your mistakes larger? You can do that with this technology, and we're having fun with this. It's a real trip. The patient and the therapist sit side by side in this example, and they work together. The therapist decides what the movement is, and unbeknownst to either of them, the patient and the therapist, they're trying to move, and there may be error augmented feedback. Either the robot is pushing them to make theirs larger, or it's actually visually distorting the image so that they see a larger error than they're actually making. And the brain responds to this. And we've shown in a clinical trial here, shown here in this figure on the right, that people get better faster and they get better more when they have been treated with this error augmentation treatment. So this is one example in a clinical trial that we've done in our lab. Okay, we're also working on devices. So we built devices. This is the simplest device I think I've ever built, and it's a network of springs programmed in the springs themselves that can actually do whatever you like to set up a torque field. So we're working on this as well, and you can see that what we have in this example video here is, whoops, I'm not even showing it to you, am I? I have to advance the slide. This example video, we can show that this network of springs is actually developed to balance the arm against gravity no matter where you move your arm. This is one example of what you can do with this, and there's many more. So with that, I'll say, I'll just wrap things up by saying that our laboratory is a nice environment where we have a lot of new technology and capabilities and the ability to get access to patients if you'd like to do research on them, and do research that actually asks questions about how the brain controls movement. Okay, and I think I'll stop there.
Dr. Patton, that was very cool. So our next speaker is Dr. Alsberg. Dr. Alsberg, the floor is all yours.
Thank you very much, Dr. Katani. Let me get this set up. All right, so I'm very excited to have a chance to briefly share some of the work that we've been pursuing in my lab. I'm Eben Alsberg. I lead the Alsberg Stem Cell and Engineered Novel Therapeutics Laboratory, or ASENT Laboratory. Our lab is predominantly a tissue engineering laboratory. So tissue loss and organ failure is a substantial health and economic problem throughout the world, and surgical strategies typically don't totally restore tissue and organ functionality, and so tissue engineering is a very promising alternative therapy to try to address these issues. My lab focuses on trying to understand how signals influence cell behavior, and then we try to design new biomaterial systems and microenvironments to present these signals to the cells in a very defined manner, so we can regulate their behavior and ultimately enhance the rate and quality of tissues that can be engineered.
So now what we have, I apologize, we're about 19 minutes or so over the schedule, but now we have breakout rooms for the faculty who have already presented. They're gonna be in their breakout rooms. You received an email from me with all the various links. If you're interested in talking to any of these faculty, please feel free to join those breakout rooms. However, in addition to those breakout rooms, we will continue talks by faculty members who want to showcase their research and sort of show you all the great things we're doing at UIC. So our next speaker is Dr. Zhangli Peng, and he will tell you about his research in microfluidics and other areas. Dr. Peng, the floor is yours.
Hi, everyone. Let me see if I can start sharing that right. Okay, let me start. So my lab is mainly about multi-scale modeling. So we also we are part of the Center of Bioinformatics and Quantitative Biology. So here is the members of my lab. We currently have 4 PhD students, then 2 undergraduate researchers. Then our formal graduate student, for example, Sam went to Hui Jie went to industry in Chicago. Then the other 2, Sebastian is postdoc at Duke, and Wei Tao at Carnegie Mellon. So we are still looking for very highly motivated students, especially with solid background in physics and mathematics. We do not do experiments. We only do computational modeling. So I will briefly introduce what we do and give 2 examples. So here we use actually my lab is called a multi-scale modeling. We also, for the hardware we are using, we kind of also multi-scale. We're using the supercomputers in the Argonne National Lab and NSF. For example, we get many millions of hours in these machines, supercomputer machines. Then we also have our own HPC computer node at UIC campus, so we can run many intense simulations and plus some HPC workstations. So let me show you one example. Computation can be very important. For example, this is a simulation of a car crash from small overlap impact. For example, you can help the car designer to make it safer. It's really important because actually turns out this is my car, happened last year. So it helps save lives. However, this is for kind of traditional hard materials. Our challenging is whether we can predict similar behaviors for kind of soft biological material like a cell. Here is the red blood cell and pass through a very small constriction. So I will show you more about this example. But there are challenges when you go from this car to biological materials because it's very soft. And there are many challenges. For example, there are very large deformations for soft material, and there are dynamic assembly in microstructures, mesoscale structures. Also, sometimes entropy dominant in many cases. So most importantly, the challenge is how to integrate different information from different scales. So for example, this showing a way to solve the electrostatic equation. Actually, different method shows much different performance. Some good method can be millions faster than a bad method. For example, the multi-grid method is kind of a multi-scale modeling approach. So additional, in 2013, actually, Nobel Prize was awarded to multi-scale models in chemistry to model these chemical systems. Why it is important? Let me give you another example in biology is sickle cell disease. Sadly, actually, we received an email last month from the biomechanical engineering say that one of their graduates is a very excellent student, died of sickle cell anemia last month. So how this happens, and these patients have a single mutation in the gene. Actually, this causes a change of the protein, just one side change, then will cause the protein to polymerization and cause the cell to become more stiffer and sticky. Then your blood rheology changes, then will cause vessel occlusion crisis in your body. Then we need to solve this kind of problem. I mean, experimentally, you need different techniques like different even microscopes, but computationally in our lab, we apply different methods, different scale, including continuum modeling like finite element, dissipative polydynamics like coarse grain modeling and molecular dynamics, and to couple all this together to understand this type of kind of mechanics related diseases. So if you want to learn more about my lab's research, you can go to the lab website. You can look at the description of my research and the recent published papers on different topics. So let me briefly describe two examples. The first example is we study red blood cell a lot. This is a red blood cell. It has a beautiful shape. When it has body, it's like a bullet, also highway traffic. So it's not only beautiful like a donut, I always say, but it's also very strong. It can pass through very small pores in the spleen in our body. So, but this is another image to see how the deformation is so large when the cell passes through the spleen. So to understand that, we also want to know that there are many cases, even in malaria, sickle cell disease, the red blood cells are not like a donut-like anymore. Then this actually the cell cannot pass through the spleen anymore. Then will block your spleen, then cause enlargement. You need to remove that. To understand, we work with experimentalists. For example, the experimentalists do the simulation using microfluid, or do the experiment using microfluid to mimic this process. Then we conduct simulation to find out the details for healthy cell, for sickle cell. For example, they have very interesting shapes, and for some anemia cells, they can be damaged. How do we model that? Actually, we adopt, that's why we call it multi-scale modeling lab. We adopt a multi-scale modeling approach. We start from the protein because then we can do all atom and atomistic simulations. Then from these atomistic simulations, we can and get information to construct more coarse-grained model at a larger scale. This is a cytoskeleton of the red blood cell. Then by simulating this one, we can also simulate the whole cell in the fluid channel, then compare with the experiment. Because in reality, it's some single mutation happens for this protein, then cause this arrangement of the cytoskeleton, then changes the cell behavior in these spleens. That is one, I think, a good example shows how we can use some modeling starting from the protein, the atoms, to understand the whole cell behaviors. So recently also we developed analytical models of this cytoskeleton mechanics, was recently published in Biophysical Journal with Stefan, my graduate student. We are also trying to extend this kind of multi-scale modeling approach to different structures, like the nuclear laminin actually has a similar structure with the cytoskeleton in red blood cell. We can study the detailed, for example, atomistic level deformation of the lamins in the nucleus. Then we can study the network. Then the problem we want to study is that one is the cancer cell migration, how the cancer cell squeeze through this kind of constrictions. And the other example is leukocyte, the white blood cell also needs to squeeze through some very small pores. Then the nucleus will be the main barrier. So actually there are a lot of microfluidic experiment on this showing that a nucleus deforms a lot. And my student, for example, we are trying to work with James Lee in our department, do AFM on the nucleus, also simulating when the nucleus passes through these constrictions, how the laminin, in many diseases actually have a mutation, the laminin impact this process. So the next example is more computational engineering. What I showed you is study the fundamental science in biology. But in engineering, for example, we help design microfluidic channels. For example, I will work with Dr. Popovsky group at our department to simulate how the particles moving at the fluid channel under inertial force. They use the high-speed hammer, but we're using our fluid structure interaction algorithm. We can identify what's the mechanism and why it's moved to certain position, but also understand what's the detailed trajectory to understand the behind the mechanisms. So my students, new student Moyin, is working on this topic for like a separation of cancer cells and other biological samples using inertial microfluidics. That's in microfluidic. We even work for nanofluid in an even smaller scale, for example, nanopore sequencing of DNA and proteins. My student Sebastian actually using molecular dynamics to simulate how the DNA passes through the nanopore, how it will cause different signal difference, and what's the like ion concentration change around this DNA. So finally, I mentioned that the beginning in the sickle cell disease is a devastating disease. So recently, we also worked on that using rheometer to measure, for example, if you change the red blood cell mixed with certain healthy red blood cell with certain rigid sickle cell or rigid Bs, how is it change the rheology of the blood? Then we worked with Howard Stone in Princeton. Then we did a simulation. For example, the blue one is the sickle cell, and the red one is the healthy red blood cell. We try to understand actually how different interaction between fluid and hard cell fluid and soft cell contribute to this kind of called shear thinning behaviors of the sickle cell blood in sickle cell patient, and to help understand if the patient needs blood perfusion, what's the percentage might be optimum for their blood have a good rheology properties. To summarize, here is a plot to summarize the research in my group. So we fundamentally, from the method of point of view, we focus on multi-scale modeling, coupling continue with atomistic modeling. Then we're especially interested in modeling the cell structures. And on the other side, we also are interested in the engineering side of the fabricating microfluid device using different physics to separate in cells and manipulate cells. Finally, I want to say we are a purely computational lab, so we worked with a lot of experimental group at the UIC or in United States or in other countries like France. So constantly exchange data and compare with the experiment, try to understand the experiment, also sometimes even guide the future experiment. So that's my introduce of my group. Thank you very much.
Thank you, Dr. Peng. Okay, so as As I mentioned before, there are breakout rooms going on right now. I sent you all the Zoom links to all the attendees, so please feel free to pop into those breakout rooms. But last but not least, Shujun Lee is going to be presenting the research of, Shujun Lin, I'm sorry, will be presenting the research of Dr. Klatt's lab. Shujun, are you here?
Yes. Hi, Professor. Can you hear me?
Yes, we can hear you, and we can see the presentation.
Okay, great. Hello, everyone. This is Shujun Lin from Motion Encoding MRI Laboratory. On behalf of Dr. Dieter Klatt, I will briefly talk about several projects in our lab. So the first is my major projects in my PhD program, which is simultaneous acquisition of diffusion tensor imaging and magnetic resonance elastography. For those who are not familiar with diffusion tensor imaging and magnetic resonance elastography, I will give you a brief description of these two techniques. So the diffusion tensor imaging is a MRI technique that is able to analyze, to assess the anisotropic property of protons in tissue. The magnetic resonance elastography is able to assess the mechanical properties of tissue. Since the nature of the acquired MRI signal is in complex form, so which means it has both magnitude and phase. Some of the MRI techniques, such as DTI, would use the magnitude as their image contrast, while the MRE mostly use the phase as their image contrast. So we thought, why not use both the magnitude and phase at the same time? So we will have immediately co-registered DTI image parameters and also the MRE image parameters. So in DTI MRE, we firstly acquire the B0 without vibration and without the motion encoding gradient. We have 24 directions that is evenly distributed in the hemisphere 3D space, along with its negative pair. We then group the 24 directions into 8 groups. Each group will have 3 directions that are as linear independent as possible. For example, if we look at the left bottom, there's an illustration of how the encoding gradients are grouped in space. Here we only show 12 directions because the 24 directions along with its negative pair would be too much to illustrate. So next, the DTI MRE sequence is implemented onto a human, like a clinical 3D scanner in our collaboration site at Urbana-Champaign. On the right-hand side, the image shows the experiment setup. The volunteer will be lie down on the bench. The brain will be located at the center of the magnet. At the back of the brain, there is a pillow. Excuse me. The changing pressure in the pillow would generate a nodding motion of the brain. And this is the results from our pilot study. We compared the results from the DTI MRE with the conventional DTI and conventional MRE. We found a really good correlation between the image parameters from both the DTI MRE to the DTI and DTI MRE and MRE. Moving on, I will talk briefly about a project that is from our lab alumnus at Xuan Machander. On the left-hand side, the image shows how he handles the animal holder and deliver it into the bore of the magnet. On the right-hand side, it shows the which is similar to the clinical scanner, the MRE sequence from the computer that send out a TTL signal to the waveform generator that like asking the waveform generator to generate a sinusoidal or trapezoidal signal that is being amplified and then delivered to the actuator. These are the results from his studies on the Alzheimer's disease mouse model, comparing to the non-transgenic control at 6 months. As evident from the image itself, both the absolute value of the stiffness and the phase of the stiffness are lower in the Alzheimer's mouse model. The image on the right-hand side shows data acquired from mice which were fed on a special diet to develop fibrosis in its liver. The fibrotic liver is more stiff compared to healthy ones. But please notice that we use a different vibration on the liver comparing to the mouse. We use acoustic waves that is transferred through a hose that at the end of the hose there is a thin film that would deliver the vibration to the liver. Please also notice the stiffness in the AD mouse are softer than in the control mouse, while the stiffness of the hepatic fibrosis are harder than the healthy subjects. The next is from another lab anonymous, Ralph Rater. He majorly used the tabletop MRE to study the native and lysed specimens. The tabletop MRE is a low-cost method for assessing mechanical properties of small tissue specimens. The frozen tissue banks potentially facilitate access to larger amounts of well-preserved specimens. However, the evidence about changes of rheological tissue properties through freeze-thaw events is currently limited. So the aim of his project is to characterize changes of fresh ex vivo, also known as native, and frozen and thawed, which is known as lysed specimen of porcine muscle, liver, and kidney tissue. On the right top, the porcine kidney specimen scanned in the axial orientation in the glass capillary. The magnitude image shows a homogeneous tissue distribution compared with the native at the same frequency. The lysed specimen would show a shorter shear wavelength, which indicate a decrease in the stiffness. On the bottom image, there's a real part of the stiffness for both native and lysed tissues at 4 different frequencies. After freezing and complete thawing, a decrease in stiffness was evident for all tissues and frequencies. Another project of his is the prostate cancer. The aim of this project is to quantify and assess the heterogeneity of viscoelastic tissue properties of fresh specimen of men with prostate cancer using MRE and histopathology as a reference. The last slide is the people in our lab. You're welcome to contact Dr. Dideklat and me myself for further information if you're interested in our lab. Thank you.
Thank you so much. That was a very nice talk, and thank you for doing it for in place of Dr. Klatt. So that pretty much concludes our symposium. I believe the breakout room should still be open. I apologize, we went half an hour over the time, but please, if you have any more questions or need more information, please visit our website, bioe.uic.edu. All the professors' emails are there. So please feel free to reach out to them, reach out to me. The recording of the symposium, I have been recording it except for one talk. I will provide this via email to the attendees. And then, as I mentioned, there'll be a raffle done offline. I have taken a note of everybody who attended. We will put it in a hat, draw names, and 3 gift cards will be emailed to the winners. But thank you so much for attending. It was great to have you all, and we hope you consider UIC as your next destination. But otherwise, have a good night, good evening, good morning, wherever you are. Take care.